Hi, my name is Ananya Prakash,
My name is Sia Patel.
and we'd like to thank you for taking your time to listen to our presentation on the effect of natural extracts on inhibition zones in E. coli.
Yeah, so our project aims to determine the antibacterial effects of the natural extracts, as Ananya just mentioned, against the growth of E. coli through examining zones of inhibition. So we had hypothesized that the largest zone of inhibition would be displayed by cinnamaldehyde from cinnamon, followed by eugenol from clove, and allicin from garlic.
Yeah, we identified our independent variables to be the type of natural extract used, like Sia mentioned. Our dependent variable is the size of inhibition zones, and our positive control group was a synthetic standard antibiotic against E. coli ampicillin. We were particularly interested in this project due to the increasing concern of antibiotic resistance and the need for alternative antibacterial agents to slow down this trend in common microorganisms like E. coli.
Yeah, so I'm just going to dive a little bit into the background research that we did for this experiment. So we learned about the different mechanisms that each of these extracts uses when interacting with the E. coli itself. So one of the mechanisms is from cinnamaldehyde. So basically what it does is that it damages the cell wall and then disrupts biofilm production, which is really crucial for the cell to for the E. coli to grow. And then the clove has been studied in gram-positive bacteria. However, it has a similar effect where it enters the cell and causes cell lysis and uh, through the disruption of the cell membrane. And then the garlic, once again, disrupts the cell membrane and causes the leakage of crucial ions and also induces oxidative stress, which prevents the E. coli from growing.
And to reiterate, the misuse and overuse of antibiotics has accelerated antibiotic resistance, and many strains of E. coli develop resistance to synthetic antibiotics, whether it be through spontaneous mutations or participating in horizontal transmission, where they exchange genetic information, such as resistance genes. This can lead to infections that require stronger dosages, greater hospital stays, and overall greater risk. Natural compounds, like Sia mentioned, give a great alternative as their multiple pathways of targeting bacteria reduce the likelihood of resistance. So, diving deeper into our procedure, for our experiment, we prepared 11 agar plates that were inoculated with E. coli and split into five approximately equal sections. One for three for each type of natural extract, and one for the negative control, and one for the positive control. For our extracts, we soaked sterile discs into their respective extracts for 5 seconds each, and then allowed them to drip for 5 seconds each again to ensure consistency. These discs were then placed onto the respective sections on the agar plates.
Yeah, so after that, we measured the zones of inhibition using the Kirby-Bauer disk diffusion technique, which basically just finds the nearest bacterial growth and where the end of that outer disk is. So that's what we use in order to find our mean data.
Yeah, and our data showed that cinnamaldehyde from cinnamon had their greatest inhibitory effect, even beating out ampicillin with a mean radius of 2.4 centimeters. Cinnamon was followed by clove and then garlic. We then conducted statistical analysis to confirm our findings and found out that clove and garlic were statistically significant from ampicillin. However, cinnamon was not statistically significant from ampicillin, suggesting that cinnamon's inhibitory effect is not significantly different than ampicillins.
Yeah, so moving forward, we would obviously like to refine our study. One thing that we would like to do is use gram-positive bacteria, such as S. epidermidis, or be able to combine certain extracts to see if they have synergistic effects on the zones of inhibition.
Mhm. And to circle back to our purpose, this research is highly relevant in a world increasingly facing antibiotic resistance. If natural compounds like these, which are found in almost every household, are found to be as effective as cinnamon has proved to be, then we can include them in many real life applications, such as topical treatments, food preservation strategies, and many other fields. Our findings open the door for further studies in natural medicine and antibiotic resistant solutions. Thank you.
Thank you.